Thank you. Welcome and thank you everyone for attending this STEM event. I would like to thank our library IT, Association for Library Technology, and John Fitzgerald, Senior Programmer and Analyst for their tech support for this event. So the University Library is honored to have Dr. Christopher Gobler with us today to share his important work on harmful algal bloom. Dr. Gobler is an endowed chair and professor within the School of Marine and Atmospheric Sciences at Stony Brook University. He received his MS and also PhD from Stony Brook University in the 1990s. He began his academic career as professor at Long Island University in 1999. In 2005, he joined Stony Brook University as the director of academic programs for SOMAS on the Stony Brook Southampton campus. In 2015, he was named director of the New York State Center for Clear Water Technology. He has been editor-in-chief for the international peer-reviewed scientific journal, Harmful Algae, since 2018. He has published more than 200 manuscripts in peer-reviewed journals that explore the linkage between anthropogenic activities and coastal ecosystems. So let's welcome Dr.
Well, thank you and good afternoon. Thank you all for attending. Pleasure to be here and to see some new faces. And yeah, today I'm going to talk about something called harmful algal blooms and talk about one of the ways we investigate those. And before I begin, I'll just mention that there's some key collaborators here, specifically people from the Gobler lab. We have some attendees there from my lab who made it here today. And collaborators from different institutes, Sonny Diamond, Louis Wirsch, Matt Harkey, and Deepak Nanjapa, and of course funding from different federal and state agencies. So I don't know any of you, so I don't know what you know, but I can just start with the super basics. I'm a marine scientist, so I'm in the School of Marine and Atmospheric Sciences. And so when we think about marine ecosystems, I don't know what you all think about, but maybe some of you eat seafood, and so maybe that's your closest interaction with the ocean. But at the base of marine food webs, we have microalgae, also known as phytoplankton. These are photosynthetic organisms, so they're like the base, they're like the equivalent of land plants. And so it's their energy that drives marine food webs. And in general, they're very, very important. So I teach in all my classes. Half of the oxygen we breathe every day comes from the photosynthetic activity of phytoplankton. They're driving the food chain, and they're actually helping to mitigate climate change. 1/3 of the CO2 that mankind has produced has gone actually into the ocean, in large part because of the photosynthetic activity of phytoplankton. So they play a really important role in affecting life on Earth. There are thousands of species of phytoplankton in the ocean, but we know there are several dozen that actually can cause harm to marine ecosystems. And we use the term to describe them harmful algal blooms. So that's when you get the growth of one of these types of algae to a density that, well, one, it may just discolor the water and make it look like you don't want to swim. But more importantly, there's some sort of negative impact on the ecosystem. And that can be on the economy because of, say, fish kills. It can be on human health. Some of these organisms make toxins. Or it can be on the organisms within an ecosystem. And in the case where these algae actually make a toxin, that toxin can accumulate throughout the food chain and affect things in the water, even things that live in the sediment. And the most common way that humans can be affected by this is via shellfish, say, things like mussels or clams or oysters or scallops. They can filter immense amounts of water in a day, and when they filter that water, they're concentrating whatever's in the water in your tissue. And so people can be poisoned by these toxins on occasion. In addition, there's other types of harmful algal blooms that we call ecosystem disruptive, and these are events that thankfully have no effect on humans, but can be really detrimental to marine ecosystems. You're just seeing here some examples, for example, of fish kills, large populations of fish dying because they're exposed to some of these harmful algae. So just to take a slight detour now and just talk about Long Island for a moment, because the harmful algal I'm going to talk about most today, these are global phenomena if you haven't guessed. So globally, in every continent we have harmful algal blooms. But I study them here on Long Island, and I'm going to talk about one that's quite common on Long Island. And as I do that, I want to introduce the idea, or just give some background about what's happening on Long Island and been happening, and introduce the concept of a watershed, which is a pretty common concept that people think about in marine sciences. And it really is the idea that the land influences the water. And that's really acute here on Long Island, in that anywhere you go, anywhere on Long Island, you're in a watershed. And so any water that falls on land has essentially, well, I guess 3 outcomes. It can evaporate right away, and that can happen in the summer often. But for most of the year that doesn't happen, and rather it often will just infiltrate through the ground. And there it becomes very consequential for all of you. If you don't know, the water you drink every day, unless you're getting it from a bottled source, but what comes out of your tap comes from underneath the ground, right? So literally you stand above the water you drink. And it doesn't come from very far away, on average less than 3 miles away. So, some people will think, there's this term people like to use, city water. I have city water. And we don't get our water from the city, so there's no such thing. You might get your water most commonly from Suffolk County Water Authority, but again, if you are getting your water from there, and that would be the case for this campus, the actual wells that are delivering that water are not far away. So literally, we're all influencing what's going into the ground and what we're drinking. And what we don't drink also has consequences, because that water keeps going into the ground, and as it does that, it pushes out more groundwater into coastal ecosystems, where it affects what can be happening in surface waters. And if we look at what's been happening through the decades, here, specifically Suffolk County, the county we're in, we live in a county where there's been enormous population growth, an increase in the population by an order of magnitude through the 20th century, to the point where there's actually more people in Suffolk County than there are in Manhattan, more than in the Bronx, more than in Staten Island. In fact, more than almost any county outside of Brooklyn and Queens in New York State. And with that growth, there's been a big increase in the amount of nitrogen in the groundwater beneath our feet, which is our drinking water source. And a decade ago, if we saw that trend, everybody would have a different answer as to why that's happening. Why are the nitrogen levels going? And maybe you can see that trend now, and you can come up with your own hypothesis. And people would throw out sort of nebulous statements like, it must be the runoff. But actually there's been many studies over the last decade, and what those studies have all come to the same conclusion is that that increase in nitrogen is actually mainly coming from what's known as on-site wastewater, and to a lesser extent, fertilizer. But as it turns out, for 70% of the county we live in, the wastewater, that is once the water goes down the sink, down the shower, down the toilet, doesn't get treated and just goes right into the ground. Now, that's not the case for this campus, thankfully, but it actually is the case, for example, in the Southampton campus, and for 70% of Suffolk County. So literally, you have the situation where toilets are being flushed, and that water from the toilet is, as gross as it sounds, it's just going right into the groundwater, which is our drinking water source. So that's problematic, and I do direct a place called the Center for Clean Water Technology where we're working towards fixing that. But that's the fact of the matter. And when that gets into, as this illustration shows, gets into groundwater, yes, we might drink it, but also it's going into coastal waters. And every year, my lab creates this map. In fact, I'm in the process right now of making the 2022 map. And what this map shows is the extent of what are known as harmful algal blooms and low oxygen conditions that occurred on Long Island only over a 3-month period. So really, or maybe 4 months, from June through September. So a very short period of time, but what you can see is that all of Long Island is experiencing what are known as harmful algal blooms, which we'll talk about momentarily, and also what we also call dead zones, so low oxygen conditions, both of which are a consequence of overloading of nitrogen. I introduced harmful algal blooms already as being either toxic or ecosystem disruptive, and we really have both varieties here on Long Island. But that wasn't always the case. Turns out, before Stony Brook was a university, we did used to have harmful algal blooms on the south shore of Long Island. And there's an amazing map right here that probably most of you can't see, but it's a historical map. It's actually quite useful because what it shows is something I'm going to talk about momentarily, that back in the 1950s, Moriches Bay, any of you are familiar with the South Shore, didn't have an ocean inlet. And so that's why we had a bay, no exchange with the ocean, and lots of duck farms, which was actually one of the biggest industries here on Long Island, that put a lot of waste into Moriches Bay. So we actually had green tides, and we didn't have a university to study them at that point, pre-Stony Brook. They actually had to bring in scientists from another university. But the Moriches Inlet formed during the 1954 storm, called the Ash Wednesday storm. That inlet was stabilized, so that map, this is obviously well before 1954. And once that happened, the green tides went away, and we had actually an amazing 25-year period, 2.5-decade period here in Long Island, where we had the record landings of two very important types of shellfish, hard clams and Bay Scallops. In this period, particularly in the 1970s, 2 out of 3 hard clams eaten in the United States came from Great South Bay, just one little bay here on Long Island. And in addition, we had the highest landings ever of the Bay Scallop on eastern Long Island. But that changed starting in 1985. That's when brown tides began here on Long Island, one type of harmful algal bloom that I'm actually going to talk the most about. And we've been in a situation where there's been all these other harmful algal blooms showing up year after year, oftentimes to the detriment of fisheries. But again, I'm going to focus mainly on brown tides. Turns out, just a little bit of personal history for myself, when I was quite young, I think probably 19, I learned about brown tides, and from a bayman who said to me that, oh, well, I used to harvest hard clams, but I can't do it anymore because the brown tide killed off all the hard clams. And, my son was going to be a bayman, he can't do it either. And I asked him, I said, well, why do we have brown tides? He said, nobody knows. But it's happening every year, and it's wrecking everything and a whole way of life. And I had actually family that had been involved with clamming in the South Shore, so it hit home to some extent. And actually they then figured out, well, this is something I want to study when I finish my biology degree at the University of Delaware. And so I looked, and there was actually only one place you could go to study brown tides, or do graduate research on brown tides, and that was here at Stony Brook. And so that's what brought me back home. I'm actually Long Island native. And that's why I did my graduate work here at Stony Brook. But anyway, back to the organism at hand here. This is what a brown tide looks like, and I just said this in my class. If you were to put your hand into the water during a brown tide, as you put it in, you'd see your fingers probably disappear by the time you got to your elbow. It's really, really turbid. That's 1 billion cells per liter, a lot of what they call light attenuation. And I mentioned in this timeline here that they showed up in 1985. What's super interesting, is that when it was first discovered in 1985, no one had seen this organism ever in the history of science and humanity. So this organism suddenly shows up, the water's brown, they look at it under a microscope, nobody knows what it is. They use special microscopes like a scanning electron microscope or a transmission electron microscope, and they see the cells. People still don't know what it is, so it had never been seen. The conclusion now, decades later, is probably was sort of lurking in the water, but it's so small, 2 microns, that no one really noticed it or cared. It was just sort of in what we'd call the background flora. So it definitely was somewhat of a niche organism when first discovered. the blooms happen simultaneously here on Long Island, both in the South Shore and the East End, but also in Rhode Island and also in Barnegat Bay in New Jersey. And then they sort of spread south through the United States, showed up in South Africa suddenly. In the 1990s, a sister species was found in Texas in the 1990s. In the 2000s, I had colleagues in China who invited me over to show me data and say, we think we have brown tide here. As I just explained in my lecture, my career is along in me saying, that's ridiculous, that's impossible, and then things actually being quite possible. In fact, not only is it the same, it was the same exact species as we have here, and interesting, in a bay that actually also imported the New York Bay Scallop. So there may or may not be a link there, but and then in the last decade we saw the more subtropical species spread throughout the Caribbean. Focusing on the species at hand here, we've seen the cell as far north as Maine, as far south as Florida, but as I sort of indicated already, the blooms have really been isolated to the northeast and mid-Atlantic zone, and they've been very destructive here in New York. I mentioned before the Bay Scallop fishery, shown down here, was one of the biggest fisheries in New York during the 60s and 70s, collapsed entirely. With the onset of the brown tide in 1885, brown tide sort of went away for a little while in the early 90s, and that fishery, as you can see, began to recover, but then the brown tide returned, and that was the end of the Bay Scallop fishery. Unfortunately, it went through the same sort of iteration of recovery and collapse again in the last few years, but that's another talk. I mentioned that the hard clam was the biggest fishery. New York was the biggest fishery for hard clams in the U.S. The original decline of that was due to overharvesting, but repeated brown tides prevented that from then coming back, and the extreme turbidity leads to the die-off of seagrasses, which are a really important habitat for small fish. So a lot of what I've done through the years, and it's kind of almost embarrassing to think about how many decades, but through the years have been to try to understand how does an ecosystem change from when you look in the water, there's lots of algae in there that are supporting a fishery and it's a healthy ecosystem, to one where you've got one of these harmful algae that's sort of co-opted the ecosystem and leading to negative ecosystem impacts. One of not one of the first things I did, but one of the more exciting things I did early in my career was actually sequence the whole genome of this organism. And so we had a whole group together. We called ourselves the Oriococcus Genome Consortium, and that was supported by the Joint Genome Institute of the Department of Energy. This was actually the first harmful algal bloom to have its whole genome sequenced, and through that we figured out a lot about the organism just by seeing what its gene set was like. And I won't go into too many details, but just give some of the highlights here. We did something called Comparative Genomics. At the time there weren't a lot of other genomes available to compare to. But even at the time, we did have at least 6 or 7 other species that coexisted with the brown tide in these coastal environments. So we could compare those genomes and say, well, what makes Oriococcus, the brown tide, different? So one of the things we found out is that it actually, despite the fact that it's so small, 2 microns, actually had a bigger genome than many of the other competing species and more genes. And so we think the larger gene set gave it the flexibility to adapt to different environments. And so specifically, and I'll get more into the details, but adapting to a low light environment, I already showed you how, and explained to you how turbid the water is and how that can be detrimental to seagrass. These are photosynthetic organisms. They have to somehow get by, and if the water is super turbid, how do they do that? Well, they had genes, more genes to adapt to low light, and also genes for using organic matter for growth. So most phytoplankton will use what we would call inorganic nutrients, so things like nitrate or phosphate in the water, which actually can be at very low levels actually during a bloom because these blooms get so intense. But this organism is able to do other things, specifically use organic matter. And then finally, we found specific genes in the organism that seem to be coding for what we call a predator defense response. So this organism wouldn't get eaten, it would express these genes to make sure that it could continue to live. So that's just the genome. But I don't know how many of you have a background or knowledge of genomes and molecular science, but the genome only tells you so much, and so really what becomes important is the genes that are actually expressed. Right? We all have big genomes, but they're just sort of sitting there. They're somewhat static. I won't talk about epigenetics, which is the special case for them being static. But in general, you're born with your genome, but what's actually important is what genes are active and actually encoding for proteins that then dictate how you or any organism is responding to the environment. So that was partly the goal of what we wanted to do next, is to look at not the genome, which is just sort of sitting there, but the expression and action of those genes. What genes are being turned on in a specific environment, and can those genes that are being turned on and activated tell us something about this organism and how it's proliferating to form these harmful algal blooms. So one of the first forays into doing that is to grow the organism in a laboratory setting under conditions that we often would see during a bloom. So under low light, where it would be low in nitrogen or low in phosphorus. And these are growth curves, but the particular numbers on the right-hand side there is their growth rate. So, and specifically the growth rate, what we called harvesting the cells, right? So this is how they grew over a week-long experiment. But as it turns out, gene expression is very, very transient. So the genes you're expressing now, you might not be expressing even like 10 minutes later, right? So you need to actually get a quick snapshot. So what we did in this particular experiment is understand what are the genes that are being expressed at the exact time of harvest. And the one that's called replete, that just means that they had plenty of light, plenty of nutrients. So it's sort of what we'd call the control condition. So in doing this, what we were able to do is come up with little maps of the specific genes that were being expressed by the organism to adapt to a given environment. And so I won't get too far into the weeds here, but suffice to say, under low nitrogen conditions, so this is supposed to be, the box here is actually supposed to be the cell. So this is like the inside of the cell, the outside of the cell. And these little circles on the outside of the cell are what we call transporters. So nutrients being brought in and out of the cell. And so we saw specific transporters turn on under low nitrogen conditions. We saw certain intracellular activities being turned on to break down organic phosphorus under low phosphorus conditions. And then we saw specifically some rearranging of lipids internally under low light conditions. And that was just in the lab, and so what we really wanted to know is does this continue in an ecosystem setting? So what about if you were to go to Moriches Bay, which you're looking at here, or Graytail Bay, where these things occur in an ecosystem setting, do you see the same sort of patterns? And so to do that, we're specifically just collecting water, and then specifically looking at the RNA for Oriococcus, which is the brown tide, and sometimes in competing species as well, although I don't think I'll talk a lot about that today. So I'm going to show a series of these sorts of plots, and they're just simply what we would call a time series plot. So you can see the time frame on the bottom axis there, going from spring into summer, and in the brown are the cells, the amount of brown tide that you see. And then specifically here, we're also looking at the amount of phosphorus, orthophosphate. And so the pattern seems to be, when the bloom is starting out, at the very beginning, the phosphate levels are low, and then they sort of ramp up thereafter. And in looking at specifically the transporters that bring phosphorus into the cell, what we saw is very high levels of expression when the levels of phosphorus are low. And then, as the phosphorus levels decline, that gene is actually turned off, right? So part of the point here is now we're identifying within the cell an indicator of the current status of the cell. So we can understand by just looking for the presence or absence of this gene, maybe we look at it quantitatively, does the cell have enough phosphorus? This is the same cell, same gene that came up in the lab experiment. So we proved in the lab, low phosphorus, it ramps up this gene. And then now we're showing in the ecosystem setting the same thing. So now an opportunity to track that gene and to see when it comes up, to use the presence or absence of that gene to literally, I'll use the term, interrogate these cells. So does this cell have enough phosphorus in an ecosystem setting? Well, let's look at the genes and have it tell us that. We further did some experimentation to try to understand how nitrogen and phosphorus would affect these cells. And here's a very simple experiment we do all the time in our lab, where we collect water and then amend it with nitrogen or phosphorus or both compounds. And again, to understand what's controlling these blooms. And just to step back for a moment, if you want to stop these blooms, well, how would you do that? Right? And so one of the approaches would be to limit the amount of nitrogen or the amount of phosphorus going into the estuary. Either approach is very expensive, and either approach and the sources of nitrogen and phosphorus are very, very different. Right? And so just because you address wastewater, that might address the nitrogen, but it wouldn't necessarily address the phosphorus. So we really need to know this in order to stop these events. So anyway, in this particular experiment, we added nothing or those different nutrients, and what you can see, firstly, just from a growth response, this is what's shown here. They were actually dying off in the absence of phosphorus, but given phosphorus or nitrogen and phosphorus together, they grew. And so, in tandem with that, we also, and I'll just pull up 3 of these up, made these plots that are probably a little complex, and so I'll walk you through them. This is the gene expression patterns in all those treatments. And specifically, you'll see a blue line, which is actually made up of little dots. Each dot is a gene. And then we can look at dots that are either above the line or below the line, because these plots are comparing the control condition to each treatment. So if it's above the line, that's more expression. Below the line, that's less expression. And so what we see with the nitrogen, not a lot of response, just a couple of genes that turned on. But when we added phosphorus, if you see all those dots below the line, we actually silenced a whole series of genes that were important for getting phosphorus. This sort of affirms what we found in the last experiment that I already showed you. This is actually a different data set. But that, in this particular case, they actually had all these genes turned on to get more phosphorus, but when you add more phosphorus, those genes are silenced. And then the biggest response across all the treatments was when we added both nitrogen and phosphorus, because we saw certain genes turning on and the same sets of genes turning off. Another way to look at this is to look at all the genes being expressed, not in an experimental setting, but actually in the ecosystem over the course of a brown tide. And turns out this happens to be one of the most intense brown tides recorded, actually the most intense in the last decade. And this was on the South Shore, where we had almost 1.5 million cells per milliliter, 1.5 billion per liter. And you can see the time course beginning in June, peaking through July, and then done by the end of July. So, in parallel, well, there's somebody's phone. In parallel, we used approaches to look at all the different types of algae in the water. And so what you can see is that there's a good mix of phytoplankton before and after that peak of the bloom. But during the bloom, most of the cells out there, 60 to 90%, are specifically Oriococcus. So it's really taking over the ecosystem. Okay. So this is going to be a little complicated. Just for fun for me, how many people here have taken a class in molecular biology? All right, so we've got some people here. But it's good to know for my audience, so so this is what we would call the global transcriptome of this organism. So all the genes being expressed by the organism on different sampling dates, and it's a nice color-coded heat map. So the superabundant genes in the lightest colors and the least abundant genes in the darkest colors, everything normalized to the full data set. Anyway, I won't go into the weeds here, but I'll just simply say that we could, if you squint your eyes, you can maybe see 3 general sets of expression patterns here. And you'll also see that the bloom is growing at a super rapid rate in the very beginning. This is quantitatively its growth rate. And then sort of a leveling off in the middle, and then obviously a collapse of the bloom. And so with that timing of the bloom, we also saw gene sets that all were turning on and off together. So for example, as the bloom was ramping up, we saw things that we'd expect to see, like lots of cell cycle genes and lots of DNA replication, things needed for the cell to grow very, very quickly. And then at the bloom peak, we saw an emphasis more on carbon fixation, but also fatty acid, both biosynthesis and degradation. And you may recall, in the lab experiments, we also saw that being very important. So maybe using fatty acids both to store them as an energy source, but then to rely on them when there's not enough energy because the water is so turbid. And then just a real collapse in gene expression at the end of the bloom, maybe as expected. Along with this, we also saw different genes that are important for responding to phosphorus or nitrogen stress also turn on and off at different times. So the two prior data sets I showed you that showed the importance of phosphorus were both actually from the beginning of the bloom. So this is the first time looking at gene expression over the entire course of the bloom. And so what it showed is that we saw, yes, a phosphorus stress response in the beginning of the bloom, but then like we saw in that other data set, those genes being silenced, and actually a bigger response for nitrogen genes during the peak of the bloom. And then nothing going on thereafter. I think I'll just pass by that. We wanted to complement this sort of picture with the same kind of experiments we talked about before, and conducting experiments at each of those different phases. So the beginning of the bloom, the peak of the bloom, and then decline, again, to understand the relative importance of nitrogen and phosphorus. And again, this is separate from this. This sort of thing here is looking in the bay. So literally these samples would be collected and they'd be flash frozen within a minute of being out of the water. So this is telling us exactly what the organism is doing at any one given moment, whereas this is manipulating the environment of the organism to understand how it might respond. But similar to the experiments that I showed before. So just to walk through these, again, paralleling what we found before in the very beginning of the bloom, and I'm not even going to worry about those too much. We saw a phosphorus response, and those little dots are just saying that each of the treatments that we used there are different. We looked at different gene sets that will respond to phosphorus, and essentially, consistent with what we saw in the experiments, many genes responding to just phosphorus, many more even responding to both nitrogen and phosphorus together, so consistent with that last experiment we saw. But together saying that at the beginning of the bloom, it looks to be a strong role for phosphorus, maybe secondarily nitrogen. At the peak of the bloom, again, consistent with what we saw in situ, just probing the cells individually, we found that nitrogen was very important for one, stimulating the growth rate of the organism. And the dot plots also showed us between the different treatments strong separation from nitrogen. And the strongest gene responses amongst the different classes of genes in the nitrogen, much more so than in the phosphorus. And then finally, by the end of the bloom, there seemed to be some co-limitation of both types of nutrients, and because the bloom was in its collapse, the gene set response was fairly muted. So overall, just maybe lingering here, I mentioned before that maybe one of the reasons we would use these molecular tools, I mean, one, I will say this is like new information and sort of the idea of understanding what an organism is doing in an ecosystem based on its gene expression response is actually fairly new in any science, any ecosystem science, but even especially in oceanography. So it's just of interest to understand what the organism is doing. But this does have practical applications as well. Some of the science is telling us that actually phosphorus is very important in the beginning of the bloom, but nitrogen is very important in the end, and we actually probably need to be managing both nutrients in order to keep these blooms in check. And that's, new real-world information that's now being implemented. There's been a big push to restrict nitrogen loading into estuaries to protect the health of these ecosystems, and just in the last year now the state is actually also putting a big emphasis on phosphorus. Okay, So here's the slide. I think I already presented in my class. I've got some of my students in here. Pretty simple equation here. That really, the thing I want to emphasize is that the nitrogen and phosphorus dictates the rate at which the organism grows. But just as important to the population dynamics is the rate at which it dies, right? And so I told you that when we looked at the genome of this organism, we found that it had a whole series of genes for making what we would call secondary metabolites that could be anti-feeding genes. So that is making some sort of compounds that would make this organism unpalatable to predators. So we wanted to test that, and we have tested that, first in an ecosystem setting. And so here's the same experiments that I've showed you many times. There's the nitrogen, the phosphorus, both things together. And now I'm showing you the treatment, well, that wasn't in the last plots, but I'm showing you here, and that's adding a zooplankton, or a grazer, so a small, something that's a predator of this cell. So you can see there it is, Oxyris marina, so it's a little flagella that swims around and loves to eat little cells. And in this particular experiment, we quantified the amount of brown tide in red, the growth rate of the brown tide, how quickly it's growing, and then also quantified the growth rate of what it's competing against, another type of phytoplankton, Phytoplankton. And so what you can see is when we added the grazer, what we'd expect is that the growth rates of everything would go down, because the grazer's just going to feed on everything there. But actually what we found is that it went after that Synechococcus cell very, very well. And you can see that's a negative growth rate, that means it's dying, exactly what you expect. But curiously, compared to the control, where we added nothing, Orchicoccus was actually growing faster. Right? So it wasn't that it was, not only was it not being eaten, it grew faster in the presence of this predator. Suggesting that, stepping away from the nutrients, if you just compare the control condition, where both organisms are growing at about the same rate, introduce a predator, and now this guy has really found an advantage. I mean, the advantage to where it, not only is it growing faster, but its nearest predator, or its nearest competitor, is dying off. This is a sign that this is probably a very important ecological process. Not one we can control, but is going on. So we actually did the gene expression work in those experiments also. These are called Manitowoc plots. Sometimes people also call these volcano plots. They're just dot plots. All you really need to look at is look for the red dots. Right? So these are all, in each of these, we're comparing the gene expression. Above the line is an increase in gene expression, or a gene that's expressed at a higher level. Below the line is a gene expressed at a lower level. And again, just look for the red dots. And what should jump out to you is that while there are some red dots that show up when you add nitrogen or phosphorus or both things, there's a lot more of them when you add the grazer. And so, you know, I look at this, and the first time I saw it, I said, these cells are going crazy, right? They're all these genes being turned on and off when the grazer's there. So clearly, this is having a big effect. And I showed you some of these sorts of dot plots before, and I skipped over them, but I'll just highlight this one. When dots are close together, they're aligning similarly. This is a principal component analysis. And so what you can see is the response to the zooplankton, the grazer, is totally different from all of the other treatments. And you've seen these plots before as well, but again, similarly, when you compare the response, the gene expression response to that zooplankton, it swamps everything from nitrogen and phosphorus. And when we look at specific sets of genes, we see whole sets specifically related potentially to making toxins or biolactic compounds and to adapting to low light that are very strongly expressed, and other ones that are being turned off. You know, again, much more so than these other environmental variables. And there's a whole suite of genes in there that may be, those are just a list of some of them, that may be related to predator defense. And of those, I guess I'll highlight membrane attack complex. And that sounds like if you want it to be if you're engaged in microbial warfare, being able to attack a membrane of a competitor or predator might be a good thing. So to follow up on this, we recently did some lab experiments kind of mimicking what we did in the ecosystem setting, where we're exposing the brown tide that we actually can grow in the lab as well. So we've got the option to do it in the lab or do it in the field. And we did that with the same grazer, but did it in two ways, either what we call direct grazing, so just putting them all in the same vessel and have those grazers have at it. But in the second iteration, the indirect grazing, we're actually most interested in the cells below that line. So we had a little membrane that would separate out the top population, the bottom population. And to answer the question is it the grazing itself, or is there some sort of, we call it compound, we call it an infochemical. And so we now realize that, we think of microbes as simple organisms that obviously don't talk, but actually they do talk to each other via sensing different sorts of chemicals. And they can talk across species, we're now learning, and this would be within species. But the question is, are there compounds that are going through that membrane that they're picking up on for the response? So here's just an example, same sort of plot. And again, I told you when the dots line up, they're all quite similar. And so you can see, firstly, you do get separation of the different treatments. Secondly, that even the indirect, that's the filtrate grazing, was different than the control treatment, but the direct grazing was very, very different. And again, similarly, we see that these are the patterns of gene expression across different pathways, gene pathways. Here I've thrown in that we had two levels of brown tide, a high level and a low level. But what I'll call your attention to is that the biggest response is when we had high levels of brown tide and direct grazing, and that the strongest response of all the different gene sets was specifically in the, you can't read it here, the biosynthesis of secondary metabolites. So and secondary metabolites are compounds that they are not making to grow, but for other purposes, probably to defend themselves. So lending to the hypothesis that yes, indeed, they do, one of the reasons they're able to form these blooms is by arming themselves with different compounds that make them less palatable to predators, and therefore the predators avoid them and go after their competitors. Okay, I think the last thing I'm going to talk about is why are these organisms harmful? And so this actually flips the script a bit in this presentation. I've already mentioned that the crash of the Peconic Bay Scallop was originally due to brown tides. And so the last thing I'm going to talk about here is actually now doing the same sort of gene expression study, but instead of looking at the brown tide and saying, well, why are you forming a bloom? Now we wanted to look specifically at the scallop, right? Even though we know that we've known for decades that the brown tide is harmful, but unlike, frankly, nearly every harmful algal bloom that's studied on the planet, we don't know exactly why this organism kills off shellfish. So in this particular study, the goal was, can we look at the organism that is affected, the scallop, look at the gene expression patterns of that organism, and understand, or try to gain insight, why is this organism dying? And so this is, it was a collaborative study from different people in the lab. It was published just a few years ago, 2019. And so just a quick note about the Bay Scallop. We've talked about it already. But it's a pretty sensitive organism. It also doesn't like really warm water. And in fact, the population is now doing poorly because we think, because our waters are getting too warm in summer. And they're also threatened by something known as ocean acidification. And harmful algal blooms is the other potential threat. So in this particular experiment, maybe you can tell I love doing experiments in my lab, we exposed the scallop larvae, so this is the earliest life stages of the scallops, to different environmental parameters: acidification, higher temperature, the brown tide, which we talked about since I started this talk, that was. And also another type of harmful algal bloom known as Coccidium or Margolepidinium. And let these scallops grow out over a 10-day period, and you can see that here. Now scallops, in general, bivalve larvae, we think in an ecosystem setting about 1% of them survive. So you always will get these sorts of declines. I teach my ecology class. These are definitely R-selected species, so bivalves will make millions of larvae, and with the hopes that maybe 1% of those millions survive, and if they do, the population's doing great. So you can see in the gray, we're getting about 50% survival. In the yellow, the temperature, well, didn't really have that big of effect. We probably should use slightly higher temperatures. But we saw a slight effect from acidification, but saw a very strong effect from both of these harmful algal blooms, to the point where, as you can see, very few of the larvae even survive. For this experiment, instead of waiting to 10 days, because if we didn't wait to 10 days to look at the transcriptome of this organism. If we did, there wouldn't have been any larvae left. But rather, we harvested them just in about 2 days. Again, they've been exposed for 48 hours. What can the organism tell us? What genes is it turning on in response to each of these different stressors? Well, here's one way to look at that. And this is strictly, these are obviously Venn diagrams, and these are strictly comparing the different treatments and the number of genes that are turning on and off. So an upregulated gene means that the organism says, uh-oh, let's turn these genes on because we need to fill in the blank, and I'll get into some of those details. Or it's saying, let's turn these genes off, we don't need them. And so what I will highlight, because I'm not sure it's going to come up in another plot, is just these two facts here. Firstly, if you look at the numbers, and just focus on what's on top, because what's in the middle is just what's shared. But if you look at those numbers on top, firstly you'll note for both of the harmful algal blooms, the numbers there are much greater than for the high temperature and low pH. So eliciting a much larger gene expression response. And interestingly, the two harmful algal blooms are eliciting responses that are exactly the opposite. So with regards to this particular harmful algal bloom, Margolepidinium, over 40,000 genes turning on. Which is enormous compared to, that was the biggest response that there was. But by contrast, when exposed to the brown tide, Oriococcus, the biggest response there was actually a downregulation. And so there was no greater downregulation in any treatment compared to Oriococcus. So the biggest gene response to the harmful algal blooms, but these two blooms giving the larvae having exact opposite responses. And that shows up here as well, although I'll probably skip over this because the details are a little bit hard to see. But just to talk now for a moment about just some of the genes. Those genes that were being turned on with response to Margolepidinium, I said 40,000 genes. A lot of them were specifically antioxidant genes. So things like superoxide dismutase, catalase, peroxidase. These are genes that if you're in a highly oxidating environment, you can protect yourself by making these enzymes. So looking like it was, and we know that actually one of the reasons that Margolepidinium is toxic is that it makes reactive oxygen species. So sort of affirming that, the scallops exposed to Coccidium are pumping out these antioxidants to protect themselves. Also putting out things like heat shock proteins, that's the one on the far right. And heat shock proteins, although they're a response to heat, they're actually a general stress response as well. And also note that in all those cases, the same genes are being expressed at a high level in Oriococcus, but they're downregulated. So they're turning those same genes off. And so what we think is happening is that there's just an overall metabolic suppression by the brown tide. And so, that doesn't give us complete insight, but maybe the idea that if you think things are bad and you're in an energy deficit, you'll actually turn off different pathways in order to survive, right? So, let's see, I guess maybe you've heard the term, if you're a starfish, the term is better to lose a leg than lose a life, right? I'm a marine biologist, so, but in this case, better to turn off as much of your energy supply as you can to survive the brown tide. That's explaining the massive down revolution. Okay, so I'm wrapping up. These are my conclusions. Congratulations, you've made it to the end. So by sequencing the brown tide genome and also looking at the transcriptomes, we've made a series of key insights about this particular harmful algal bloom. Firstly, if we look over time, the answer is that it's changing. Its response in the environment and its biochemical response is dynamic and evolves over the course of the bloom. So it looks one way at the beginning, another way at the peak, and totally different during the decline. And that the bloom seems to shift from phosphorus limitation to nitrogen limitation over the course of the bloom, and as I mentioned, that has big implications. It means if we want to eradicate these events, we probably need to control both nitrogen and phosphorus. And there's definitely signs that predator avoidance is going on, so they're able to elude predation by making secondary metabolites. And if you're not being eaten, that's another way that these blooms can occur. And while their competitors are being consumed. And then finally, it looks like the brown tide causes mortality in bivalves by having a depressive effect on their metabolism. So with that, I'll come back to my collaborators and thank you all for your attention. And if that flew over everybody's head, maybe there's no questions, but if you have any questions about the science or about science in general or anything, literally, ask me anything. Or nothing at 5 o'clock on a Monday. Yes?
Is there any consensus or agreed upon reason why in the mid-'80s, what caused this brown tide to sort of come out of hiding or appear?
Yeah, so again, refer back to my class, there's a hypothesis I love, or a saying I love from a Dutch scientist in the 20th century. It's a microbial ecologist who coined the phrase, everything is everywhere and the environment selects. And so in the very beginning, there was a thought that, this organism was transported here, it had never been here. But actually in doing research, it looks like it has been around for a very long time. And so I think something about the ecosystem changing, something changed in the ecosystem at that time to favor the bloom and favor that organism. Certainly increasing nitrogen is part of it, but there's probably also a climatic aspect because it wasn't just in New York, it was in New Jersey and Rhode Island as well. And when, once it, and it's probably both sides of the equation, both the growth and the predator exclusion, when you get a new organism that comes to grow at very dense levels, that's the equivalent to something known as an invasive species. Invasive species tend to do very well in ecosystems because predators haven't seen it before and don't know what to do with it, and will often avoid it. Yeah?
Does brown tide have any positive effects on the environment, or is it only harmful?
At low levels, its negative effects are dose dependent. And what that means is that when it's at minor levels, it's a little consequence and probably can serve as a food source to support the ecosystem. And that's probably what it had been doing before 1985. But it's once it gets to those and it's actually around year round. So probably most of the year, when it's just at, say, a few thousand cells per milliliter, it's actually probably somewhat beneficial. There may be some things that eat it. So it's really just when it gets to those super high densities that it is detrimental. Yeah?
I think you did a great job of explaining it to a non-scientist.
Okay.
So thank you. But my question has to do with that in terms of policy development. You were saying how it's expensive to kind of manage the rates of the nitrogen and the phosphorus. So how do we take this information to the policymakers and water authorities?
Yeah, on the nitrogen front, the very good thing is that this information about the nitrogen release has been out for quite some time. And there's now actually active policy in Suffolk County. I mentioned earlier on, the biggest source of the nitrogen is wastewater, on-site wastewater, and that we're literally in a situation where our wastewater is going into our drinking water. There's actually active policy now to reverse that. And so there's a plan. They've identified literally 200,000 homes across Suffolk County, which is more than half of those systems in specific areas where they're seeking to change those septic systems into advanced ones that are like essentially the equivalent of a mini sewage treatment plant, so that the waste is being treated before it gets returned back to the groundwater. And they specifically are targeting areas that are near drinking water wells, thank goodness, and also near water bodies that can be harming them. And then the phosphorus front, that's really just starting to come to bear, and so that the most important sources of phosphorus probably differ based on where you are. But in some cases, it can be sediment, like muds, and so that gets tricky. Right? There are projects that dredge muds out of the bottom of bays, usually for navigation purposes, but that's a possibility. But there's also, as these wastewater systems are being upgraded, now maybe the idea to make sure they remove both nitrogen and phosphorus as opposed to just the one. And actually, the Center for Clean Water Technology here at Stony Brook, we're looking into, what are the best technologies for it. Anybody else? Okay, well, I thank you all kindly for coming and for your attention. Have a great day.